Hello, everybody. My name is Marie, and my presentation is going to be on the electrical pacemaker activity of cardiac autorhythmic cells. And the autorhythmic cells are located in the heart, and these are the pacemaker cells. Here's a diagram of the heart, and the sinoatrial node, which is what my presentation is going to focus on, is a small area of cells that are located right where the right atrium and the superior vena cava come together. And these are the cells that are going to generate electricity that are going to travel through the atria, through the bundle of His, down through the bundle branches, and to the Purkinje fibers. And this little area of cells, if you look over here on the more detailed diagram here, these are cells that make up only about 1% of all of the myocardial cells, and it's a little crescent-shaped cluster of myocytes, which are only a few millimeters square in area, and it's about 10,000 cells that make up this sinoatrial node. And the electrical current that's generated by them is going to travel through all of these myocardial cells and through these intercalated discs with gap junctions. They are going to promote the electricity to travel through. So here is a case where structure is related to function. The action potential for the sinoatrial node cells is quite different from a skeletal muscle action potential, and it's different from even cardiomyocytes. When we look at this action potential, one of the most glaring differences, I think, is this slow depolarization that we see here in gold. And you can see that the cell never really rests. It's constantly, there is no resting potential. They display this pacemaker activity, and they actually have what's called a pacemaker potential. And it begins with some odd ion channels called funny channels. And these funny channels are allowing sodium to have an influx in, and potassium channels are closing, and they are opening when the cell is hyperpolarizing, which is different from other action potentials. So this contributes to this slow depolarization when one action potential is finished, and then they begin to open here. So if we go back again, we have a depolarization, and then we have an area where there's an outflux of potassium, and there's a repolarization coming down to what normally would be a resting potential, but it doesn't rest and it slowly depolarizes. So there's this inward current of sodium and outward current of potassium. And the other ion that's very important is the calcium channels. And there are really two types. There's a transient channel for calcium that are slowly allowing calcium to come in here. And then there are some long-lasting calcium channels that allow the calcium to influx right here as it depolarizes. And that's what contributes to a depolarization. Cardiac cells are special, and they are related to the sinoatrial node cells because they are going to have to carry that electric current. And cardiac cells have some similarities with skeletal muscle cells. As you can see here, they are striated, thick and thin filaments. They still have troponin and tropomyosin, so they're still contracting once they get the electrical impulse from the sinoatrial node. And they have mitochondria and lots of myoglobin. And cardiac cells are similar to smooth muscle cells because they're slender and short. They're about 50 to 100 micrometers long. They can initiate their own action potentials, and they have the gap junctions. The heart is innervated by the autonomic nervous system. There can be problems with pacemaking cells of the heart, and that is usually called a sick sinus syndrome. And that's when you have any kind of a malfunction with the sinoatrial node. And if the heart is beating too fast because the SA node is firing off too fast, that's called sinus tachycardia. If it is firing off too slowly and the heart is beating too slowly, that's called brachycardia. A sinus pause and arrest means that the signals from the SA node are pausing, causing skipped beats. And the other way to get skipped beats is to have an exit block. An SA node exit block signals the upper heart chambers to go slower, the right and left atria, and then you have skipped beats because it's out of sync with the ventricles. A chronotropic incompetence means that the heart rate is generally normal at rest, but it doesn't increase when it's supposed to with activity. And here is a fibrillation. Here's a diagram showing the erratic nature of the electrical impulses when you have a fibrillation, which can be tachycardia or brachycardia. And that would show up on an electrocardiogram, as shown below. Solutions for problems with the SA node include medication, pacemakers, and ablations. Usually a doctor would begin with medications. They may try things like blood thinners. Medications like Coumadin, Jantavin, and Pradaxa may be used. They may try an artificial pacemaker, which is a battery-operated small pack that is sewn under the collarbone. It's a minor surgery done within about a day. Or they may use a biological pacemaker, and bioengineers are now studying using pluripotent stem cells and transcriptomics, which involves the use of RNA and using the RNA to express certain genes. A cardiac ablation for atrial fibrillation is shown here in the diagram, and that is when they go in and use radiofrequency energy through a long catheter, and they will destroy some tissue depending on where the problem is. Here it's showing the SA node, but it could be the AV node or the Purkinje fibers down here. And depending on where it is, they may choose to destroy some of those cells, which may help. An interesting study and review paper that I found is on the impact of chronic fetal hypoxia and inflammation on cardiac pacemaker cell development. And these authors were interested in observing what fetal cell SA node cells, how they developed under the effect of hypoxia, which is a decreased amount of oxygen and inflammatory environments on the pacemaker cells, and whether they could connect that to an early detection of future cardiovascular diseases. And they explained that there are three components for this dynamic network that generates an intrinsic heart rate variability, known as HRV or IHRV. And that is the idea of looking at the SA node on three different levels. At the cellular ion channel level, as an isolated tissue level, because the tissue is under its own kind of architecture, and the fact that the energy reservoir for the SA node is brown adipose tissue rather than relying on glycogen, which the rest of the myocardium does. This is a diagram showing some of the influences on the intrinsic heart rate variability, and some of the adverse conditions could be anything from infection to hypoxia to ischemia, malnutrition, or the imprint of the autonomic nervous system. And the heart and the SA node is influenced by vagal activity and from the autonomic nervous system, the parasympathetic system, and the sympathetic system. And these are the neurotransmitters that influence each of those. So they will be sped up if needed, for the fight or flight response, or it will be settled down for a resting response. The disruption of the spatiotemporal genetic program most likely impacts the cardiac pacemaker cell synchronization, and it probably leads to a pathophysiological imprint. So they noticed that they are influenced by these vagal nerves, and each channel in a pacemaker cell has one or more gates that can be modeled. So when they looked at the SA node, if you electric shock the SA node near the pacemaker or in, in the vicinity of it, the rhythm changes, but then it reestablishes itself and it seems to have a memory. And they're calling that an imprint. So, does it get imprinted at the time of fetal development that can result in predicting what can happen in the future? And there are 12 genes involved in differentiation of the cardiac pacemaker cells in embryonic development that could be studied. Some of the other future suggestions that they had were studying profiles of biomarkers, exploring pathways that might influence mechanisms by which uterochronic hypoxia may imprint upon intrinsic heart rate variability. Are there HRV properties likely to capture it in vivo in a living system? Can we distinguish between the effects of chronic fetal hypoxia versus inflammation? And what is the relationship between CPS and myocardial development? So, the vulnerability of the fetal cells may indicate future susceptibility to cardiac dysfunction. References, and thank you.